Hi, everybody. This is Amy from Nexcelum Bioscience. I'd like to thank all of our attendees for joining us today for our Celigo user training webinar. The topic today will be how to prepare and analyze an antibody assay on the Celigo. It will be presented today by Sarah Kessel. She is our application scientist and lab manager at our San Diego office here at Nexcelum. I'd like to remind all of our attendees that you will be muted throughout the duration of the webinar, but if anyone has any questions throughout the webinar, feel free to type them into the questions bar and we will have time at the end to answer any questions you have for us. Also, the session today will be recorded, so everyone will receive a link to the recording after the webinar has concluded, and you can watch the webinar over again, or you can even pass that recording along to a friend or colleague if you wish to share the content. Okay, so I'm going to pass it over to Sarah to begin our presentation today.
Thanks, Amy. Hi, everyone. Welcome. So in today's training webinar, as you heard, we're going to do an antibody binding assay. So just as an overview, we're going to talk about a little bit of the background of the assay, why and when you would want to do this assay, how to prepare your plate for an antibody binding assay. It's a little bit different than your normal cell-based assay, and then how to acquire your image or image your plate on the Celigo. And then we'll talk more about the analysis, which is usually where people kind of get a little bit tripped up, but we'll go through some of those settings specifically and how you can customize your parameters, and then how to export your data and how to prepare your data into a graph. So without further delay, well, let's get into why and when you would be running an antibody assay. So typically, this is what you would call a functional assay for the detection of antibodies that are binding to cell surface markers, specific cell surface markers. It's most commonly used in industry for measuring monoclonal antibody production from hybridoma screens for specific protein targets. You'll also use it to identify cell populations from primary cell isolation, so what kind of immune cells are you isolating? And then cell surface marker identification, so if you're trying to identify specific markers for cancer cell lines. And then also traditional ELISA method has been used for antibody binding assays, but it has limitations. That's non-native conformations that are bound when the proteins are immobilized to plate. So it's not the natural state for the antibodies. So a cell-based assay is more functional, that it's best for looking for proper function of the antibody binding to the proteins that are bound to the surface membranes. So moving on to how to prepare your plate, it's very important to note you need to have a positive and negative control for this assay. So clearly identify you've got the right antibody bound. So in today's experiment or training session, we're going to have two cell types. One that is positive for this CD3 marker, which is our Jurkat suspension cell line, and then a negative cell line, which is the A549 adherent cell line that doesn't have the marker. Our antibody of interest today is the anti-human CD3 antibody, SK7. So this would be, and then we have our negative control antibody isotype, which is our MOPC21. These are all purchasable online. And then we'll have a secondary antibody, which is labeled with PE, so we can see it in the red channel. Then we will also use TUX as our total nuclei stain, so we can have a total count of our cells. That helps you normalize, but also identify where the stain is coming from specifically on the cell. And then we'll also need blocking buffers. Typically you use PBS with either FBS or BSA. And then we'll need a wash buffer, which can be also your PBS, or in some cases, cells are sensitive, so I'll use the HBSS. We'll need two different plate types today. It's a clear bottom, it's a V bottom plate, and then we'll also use a flat bottom black wall plate for imaging, the 655090 from Grindr. So the plate preparation actually happens first in the V bottom plate, so we can basically do spinning down and getting our cells back in pellets. So first we're going to block the cells in a tube, just a 15 ml tube if you want, for 30 minutes and an incubator. Or it can be in a flask. We're going to want to count the cells and prepare a cell suspension of 1,000,000 cells per mil. And then we're going to prepare 4 tubes labeled A through D. and we're going to be adding the appropriate antibodies, either the positive or negative antibody, into the PBS plus FBS, the block buffer. So these 4 tubes are prepared, and then we're going to pipette from the A through D tubes. Well, first of all, we'll put in our PBS, FBS buffer into columns 2 through 12. So you'll be putting in 50 microliters there, and then you're going to pipette 100 microliters out of our antibody tubes A through D into column 1 as labeled here in this plate map. So you'll see its duplicate. And then we're going to serial titrate the V bottom plates. So the content from column 1, we're moving 50 microliters from column 1 into column 2, and then also continue to titrate out 50 microliters into from 2 to 3, 3 to 4, 4 to 5, and so forth. Now on column 12, you're going to not put any of the antibody in there. You just leave that at 50 microliters. Okay, so now we have our antibodies titrated out into a V bottom plate. We're going to add the Jurkat cells into rows A through D, or A, B, C, D, and then the adherent cell line, which is actually now suspended, into rows E, F, and G, as described in this plate map below. So the top half is the cell line Jurkat cells, and the bottom half of the plate is the negative cell line A549. We're going to incubate. It's going to be 100 microliters per well. We'll incubate for an hour, and then we're going to wash 2 times with PBS by centrifuging the plate so you have a pellet of the cells at the bottom, and resuspend back into PBS, do that 2 times, and then prepare a mixture of the secondary antibody with the Hoechst, and then add 50 microliters of that to the plate and incubate for half an hour. So now your cells are stained with the antibody as well as with the secondary and Hoechst. You're going to wash this 2 times and remove any excess secondary and Hoechst. And at this point, we're going to prepare the imaging plate. So we have resuspending the pellet from the V bottom plate. We're going to take 100 microliters of that. We're going to take 50 microliters of that stained V bottom plate into 100 microliters that is in the Greiner plate, the 655090 plate. So this is basically half the amount of cells from the V bottom plate is now going into the flat bottom Greiner plate. You're going to quickly centrifuge the plates to settle the cells down. So I typically move where the centrifuge goes up to like 1,000 RPM, then turn it off. And then you're going to image on the Celigo using expression analysis, target 1, 2, and mask. So on the Celigo scan tab, you're going to select that from the drop-down menu. So it looks like this on the screen. And then you'll first want to set up the mask channel, which is going to be our Hoechst stained cells. So we'll have the blue illumination, and that usually is around 80 to 100,000 microsecond exposure. Your object pixel intensity should be between 125 and 175 with the background about 50. So just hovering your mouse over the screen, you'll see the pixel intensity displayed at the bottom of the Celigo GUI. You'll want to use the focus setup button and then select hardware autofocus and then auto registration. The system will focus and find the right focus for the blue channel. Then we're going to set up the target 1 channel for red illumination. And for this antibody, it was a little dim, so I had to use high exposure. And then we'll want to do fine focus and set offset to set the focus offset for this channel. And then for target 2, we'll use bright field illumination. This is a little fast. So for the bright field illumination, that'll be target 2. The auto exposure and gain would be just fine to use, but ideally you want to have the background to be around 125 to 175. So hovering your mouse over the background, again, you can see the values displayed on the screen. So that's the setup of the focus and illumination for the 3 channels for this image acquisition. All right, well, let's turn this back up. Okay. And then once that's set up, you just hit start scan, and then the software will start to acquire the images for the plate. And so we have already acquired these images, and now we're just going to view and analyze the images. That would be the second button down on the Start tab. And then we'll select the scan from the drop-down menu. Here we have, and this scan actually should be provided on your systems. If you don't have it, you can call us or email us, and we can send you this example scan for you to play around with if you want to check out the illumination and the segmentation settings. So from this, we select the scan, and then we're going to create new analysis. So that's the second button here. That means it'll load without any analysis settings other than the default. And we'll select an application that has 3 channels. So we'll take target 1, 2, and mask, and then load scan. So the system will move from the Start tab over to the Analyze tab. And you'll see that the application at the top is target 1, 2, and mask. These channel names are a little bit generic, so you can customize them by using this little piece of paper with a hand pointing on it. That's our customizable relabeling the channel and feature names. So for this case, I'm going to use the far red for the channel 1. And channel is the image channel, feature is the analysis object of interest on the image. The channel 2 will be bright field, and then the mask channel is the blue hook staining cells. So then click OK. And you'll see that the generic names are now customized to what we just described. Clicking on a well in the plate map, it displays the image of all 3 channels, so we can zoom in. This is our Jerkett cells. So we can turn on and off individual channels for easier visualization. So in this case, we can see that there is a far red stain, but it's very dim, so we can artificially increase the pixel intensity. This is post-image image acquisition, so I'm not changing the image other than for my visual purposes. So you can see that certain cells have some antibody bound to them. It's the bright purple that comes up on the screen. Okay. So now you can see a little bit better that the antibody staining is present. It's just very dim. And I did use quite a bit of high exposure. So ideally, if you have a better antibody, then I would use that. But sometimes you just don't have that luck. Okay, so for this application, we're going to use the nuclei to identify all of the cells in the image. So in this case, we're going to turn on the graphic overlay for the blue mask. Oh, okay. So let's click over to the edge of the well from this particular well. We want to identify the well mask so we don't count objects outside the well area. This is a black wall plate. So just turning on the well mask graphic overlay and turning on the well mask here. That means only objects inside this orange line that's going to be displayed once I click Auto Analyze. Auto Analyze is basically will auto update anytime I change analysis parameters. So when I click on that, so now anytime I make an adjustment on the left-hand column, it'll automatically update on the screen on the image display area. So the mask is a little bit bigger, so we'll just decrease it from 100 down to 98%. It really, 100% would have been fine. I think just to show you the ability to shrink it or increase it, you can use that. Now for this application, the blue mask is identifying all the nuclei, and some of the cells that are clustered close together are grouped as one, so we can separate them by clicking on Separate Touching Objects. And that looks pretty good. Additionally, you could decrease the cell diameter from 10 to 8 if you wanted to also try to separate some of the objects. But our antibody is on the surface of the cell, so what I'd like to do is dilate out 1 pixel from each of the nuclei identified area. That will help me detect some of the antibody surface staining. You can see how the graphic overlay got a little bit bigger than the nuclei originally were identified. Okay, so that was pretty much the only 2 parameters that I changed for this application. The default values, or actually that would be 3. Those are the only 3 values that I changed. Everything else seems to be pretty good. If you miss a few, it could be cells that are clustered together or too small. It's okay. Now, as you're analyzing it, it takes you to the results tab. The thumbnail images are displayed, and as the wells are completed or analysis is completed, the data is displayed on the thumbnail. So currently we're looking at the total count of hooked cells, so you can see how consistent your cell plating is. That's ideal. You want to have the same number of cells per well, so you have consistent data as we titrate our antibody out. And then we're going to move down to where our antibody of interest is. So this is the average far red integrated intensity. So it's adding up all of the pixels that are having a value in the far red channel. So our highest concentration of antibody is in column 1, and it's titrated out 2-fold. So you can see that it's pretty maxed out in columns 1 through 4, and then it begins to dilute out after that. The top 2 rows are a positive cell line with the antibody bound to the cell surface, and then we have our negative antibody that will allow us to identify the background of the RPE. And then we have our negative cell line with the positive antibody, also similar in background noise, and finally our negative cell line with the negative antibody. So these are all appropriate controls to identify what signal is real and what is background from non-specific binding. All right, once the data is displayed, you can use the heat map to help identify where your baseline is for your staining. So adjusting the min values and the mac values. As well, you can change the color of the heat map display. So this is just a temporary color change and value change. If you close this application out and reopen it, it comes back to the default values. And the default values are actually plus or minus, was it 5% and 95%? That would be the min-max for the data set that's displayed. So you can see where we have positive stain and it starts to titrate out. That looks pretty good. So at this point, we'll export the data using export well level data into a CSV file. We'll just save it onto the desktop where I actually already have a Excel template that's already prepared and ready for me to paste the data into. If you want a view of a well, on it and the high-res image shows up, and then you can also see the data displayed on the left-hand column. So this is a positive antibody stain sample. There's our blue hook stain of the jerkets. And we can turn on the graphic overlay to see that we've properly identified every cell in the well with the nuclear stain. And we've dialed out a little bit. So any pixel inside the graphic overlay will be analyzed in the red channel, and that's where the average far red integrated intensity is being displayed. So when you have a negative sample that is looking in the graphic overlay area of where the nuclei are, and then showing that pixel intensity. So you really are specifically looking at the number of cells and then the area of those cells for any positive antibody signal. So it's kind of direct cell analysis for antibody staining. You could also do further analysis to see what percentage of cells are actually presenting this antibody, or if you wanted to do intensity based, so a high percentage of cells that are high intensity or medium, that would be done in the gating tab, which we're not going to showcase today. So here in this Excel file, I've created a template. So I'm going to paste the data from our CSV file in here. This is the CSV file, and the top portion is very high level information about the plate, the scan, etc. And then below that, you have all the details of the data that were output on the CSV file. So you have like total counts, the intensity values. Well sampled as if we had done only a few images within a well. Right now we took the whole well, so it's 100%. Down below that you have the mean intensity, the standard deviation of the mean intensity, and then the integrated, which is what we're interested in today. So we'll just highlight the columns A through M and then hit Control-C on your keyboard. And then we'll move over to the other Excel sheet, which has got the template, and then we'll paste it into the column A through M. It's actually gonna, the data from that will be used in populating into this graph page that I made. Control-V to paste it here. And you can see how the data populates and the graph automatically updates with the integrated intensity showing, as well as I've plotting the average of the two wells. Those would be replicates, and then we have the standard deviation plotted as well. So now you can see where we've got our plateau on our antibody, where it's maxed out or saturated, and then it comes down and we can get our IC50 if you want, or yeah. So we have our positive cell line, positive antibody showing proper signal, and then we have our negative antibody and negative cell lines not showing any signal. If anything, that's just background noise from either the media or the secondary antibody. So that's it. So basically, to review, we've basically covered when and why you would use this assay, how to prepare your plate for an antibody binding assay, and then how to image your plate on the Celigo, as well as how to set up the analysis parameters. The default settings are pretty close, just a couple of minor changes, and then also how to export your data and plot them into a graph. So at this point, I would like to open up the forum for any questions that we can help answer for the attendees.
Thanks, Sarah. That was a wonderful presentation. So like Sarah said, we are opening up this time to answer any questions our attendees have for us. As a reminder, everyone is muted, but please type your questions into the questions bar, and I will read any and all questions out to Sarah, and she will be able to answer them for you. We already have quite a few questions that have come through throughout the webinar, so I'll just start with the first one. So it states, what if I want to test my antibodies against cells cultured and adhered to my test plate?
Okay, so the surface antibody might be a little bit bigger and beyond the nuclei. Then you could probably use the application called Expression Analysis Merge. And if you want, we can do an online session together to kind of go over some of the details on that one. So you would probably want to use a hook staining to get at the nuclei, but then also have the second channel with your antibody with like a merge. With identifying the antibody in your second channel, and then you would click the merge button, which is at the top of the left-hand column of the parameters in the analysis tab. It merges the two graphic overlays together as one, and then the analysis and the pixel intensity would be displayed from that solid one single mask merged together. Again, if you want to have some online session time, I'd be happy to help you with that.
Thanks, Sarah. Next question states, which well do you use to set up the exposure time?
Okay, so I would use the exposure time for your Hooks channel can be any one of the wells because they all are stained equally with Hooks. But for your secondary antibody, I would set up something that you know has the positive signal, positive cell line. Use that as your well for your channel setup for illumination focus for your secondary antibody staining. Great.
Another question? Why integrated intensity and not mean intensity?
Considering all the cells are the same, I kind of figured that's normalized that way. So then you can use, some cells have more pixel intensity than others. When you use mean, it's actually dividing it by this, the number of pixels within the graphic overlay. So I want to be able to pluck out stuff that's just got a few specks of antibody staining and some that have very few. So I want to have the whole picture, the integrated, just is summing up all the pixels, and mean intensity is actually dividing it by the number of pixels or the area of the graphic overlay over the cells.
Great. So our next question is kind of a mouthful. It's got a couple questions in one, so let me know if you need me to repeat any questions that I read out to you, Sarah. It starts off with, what is the advantage of acquiring the bright field image in this experiment? Can the procedure be performed on non-live cells, for example, with fixed pre-mebilized cells? And how can you improve the cell object detection when the cells are not spherical and are more flattened or have complex structures like overlapping dendrites?
Dendrites. Okay, I gotcha. Sounds more like neurons that the person's looking for. So bright field is not necessary. You don't have to image with bright field, but I know that a lot of customers just like to see that their cells are there, and visually bright field is just a good added image to have. They didn't add any quantitative information for us. It was more of a safety visualization for us. So you don't have to include it. The second of all, you can do the same kind of assay on a fixed permeabilized cell-based assay if you have that situation going on. Again, I would use some kind of nuclear stain. If you don't have Hooks, I can tell you about other nuclear stains that are available. DAPI would be another one you could use for the fixed staining, but Hooks, I think, does work with the fixing. But anyways, you can use this assay with a fixed plate. And for the dendrites, or if you have things that are going beyond the nuclei, then I would probably use, you could probably use the merge application, which would pick up things beyond the nuclei area. And I have some other ideas of some other applications you could use. So I would start with the merged expression analysis. And if you want, and again, an online session, I can work with you. And maybe from that information, I can probably maybe drum up another webinar in the future to kind of cover those abnormal cell types. So call me, or email support, and we can do an online session together, okay?
Great, thank you, Sarah. Okay, another question. What is the highest resolution and how long does it take to scan a plate at standard and highest resolution?
Okay, so our image acquisition is acquiring the plate at 1 micron per pixel. You have the ability to do it at less resolution, which in this case is not of interest for you. For one channel, it's about, it can range between 5 and 10 minutes depending on your exposure time. So for instance, in the example today, we did 3 channels, but you'll note that the antibody I had to turn up the exposure, what I consider is really high. So something that's above 100,000 microseconds for me is not my favorite. I would prefer to stay below 100,000. So that just adds more time because it's every picture that you take, and for a 96 well, that's 16 per channel times 100,000 microseconds per image. So more exposure, more time. So it's variable. But as a kind of a golden rule, what we tell people is for one channel, it takes between 5 and 10 minutes. So if you did 3 channels, figure it's about 15 minutes.
Thank you. Next question. If you were to plot the mean intensity instead of the integrated intensity, would the plot look different?
So in the webinar video, I did showcase that you could see in the CSV file the mean intensity. There was a titration out at the top end of the Jerkit positive stained antibody, and it did form a curve. I would say that it just didn't have a bigger dynamic range because it's actually divided by the area of the cells, and there's some pixels that are super bright, and that gets kind of averaged out. So I plotted in before this webinar, I plotted out both ways to kind of see what is the best way. I felt the dynamic range was a lot more obvious in the integrated intensity.
Great, thank you, Sarah. Okay, so I haven't seen any other questions come through. Thank you for all our attendees who have submitted the questions today. That will conclude our webinar for today, but if any of our attendees think of any more questions later on or after this webinar, feel free to email us at support@nexlum.com, and I'll be able to pass those questions on to Sarah, and we can email you personally and make sure that we get all your questions answered.
Also, Amy, I want to add it. Go ahead.
Oh, sure, go ahead.
So the protocol that I ran for this, we have available for you guys to review. If you want, contact your field application scientist or contact support at Nexlum if you'd like a copy of the protocol that I was running today for your review, okay?
Yeah, thank you, Sarah. That's a wonderful resource. Absolutely. Thank you, everyone, all of our attendees for joining us today, and thank you, Sarah, for a wonderful presentation. This was a wonderful session, and I hope everyone has a really great day. Thank you so much.